Covishield and Covaxin are the most widely known COVID-19 vaccines in India today. Both vaccines were brought into circulation in the first half of 2021. While Covishield is made on a viral vector platform, Covaxin is an inactivated viral vaccine. However, as early as October 2021, India may see the rollout of the world's first DNA vaccine, ZyCov-D. Produced by pharmaceutical company Zydus Cadila, the vaccine has received Emergency Use Authorization and is also the first in the country to be approved for children above the age of 12. Yet, questions abound concerning the concept of DNA vaccines. A DNA vaccine is a type of vaccine that introduces a specific antigen-coding DNA sequence into the cells of an organism to induce an immune response. How are they different from the mRNA-based Pfizer and Moderna vaccines? Do DNA vaccines have higher efficacy? Dr. Gagandeep Kang, Professor of Microbiology at Christian Medical College, Vellore, has some answers.
The mRNA platform is a platform that directly carries the message into cells about how to make the spike protein. So the vaccine itself consists of mRNA, which is wrapped up in a lipid nanoparticle, a fatty nanoparticle, which integrates with the cell membrane and then delivers the mRNA into the cell. The DNA platform is a bit different in that it also encodes the message, but the piece of DNA that carries the sequence for the spike protein is integrated into a plasmid, which is a circular piece of DNA, and that has to be delivered inside the cell. Once the plasmid is delivered inside the cell, the DNA message has to go to the nucleus, be converted into mRNA, and then come out into the cytoplasm so that the protein can be made. So there is this extra step where DNA has to be converted back into mRNA, which makes the two vaccines a little bit different, even though both of them encode the message for how to make the spike protein. Well, once the protein is made inside the cell, it can be displayed on the surface of the cell or it can be released outside. The body recognizes the protein as being foreign and responds to it by making an immune response so that it can block the protein from doing its function, which is to attach to host cells and get inside them. Now, because it's only the spike protein, that means that this is not the same as an infection, even though the immune response that is produced is at least partially the same immune response that is produced in infection. And because the spike is the functional protein which is responsible for getting the virus into cells, if we have antibodies to the spike protein, then that means that we can block the virus from getting inside cells. So that's how protection is induced when you have a spike-specific vaccine. Well, actually, not all mRNA vaccines are 90% effective. We've seen a difference between the Moderna and the Pfizer vaccines, particularly in terms of the duration of the immune response. We've also seen a difference between the CureVac vaccine, which had just about above 50% efficacy, and that's also a vaccine that's on an mRNA platform. So not all vaccines, even if they are on the same platform, are going to perform in exactly the same way. It certainly seems to depend on the amount that you're putting in. So there is a question about, in ZyCov-D, would increasing the amount of DNA decrease the number of doses of vaccine that you give? A lot of tweaking needs to be done to ensure that there is an optimal protective immune response.
The vaccine may be rolled out in India as early as this month. According to a company press release, the Phase 3 clinical trials showed the primary efficacy of 66.6%. The data, however, is yet to be published in a peer-reviewed journal or made available in public.